Terrific. Well, thank you so much for inviting me to be part of the annual convention. I'm really happy to be here. So this morning, I'm going to be talking about the work of primarily of master's student Amanda Mitchell, who revisited an earlier question that we had been studying a number of years ago. So as mentioned just a minute ago, this morning we're going to dig a little bit deeper to learn about soil organic carbon storage from Hillslope erosion-buried soils. There we go. All right, so just by way of a little bit of background, we often talk about the idea that soil organic matter is really the cornerstone of soil health. And that's because soil organic matter provides tilth, that good soil structure. It's really critical for aggregate formation and stability. It's a source of nutrients for microorganisms and for plants. Importantly, we'll come back to this in a minute, it's the primary source of energy or food for microbes. And it has critical regulatory functions. It regulates soil moisture availability and soil moisture retention, nutrient cycling, and has really critical roles in regulation of climate change through soil carbon retention and the emission and or reduction of greenhouse gas emissions. So really, at the end of the day, soil organic matter is critical for resilience. But as a microbiologist, I'm really focused on this idea that soil organic matter is the primary source of food and energy for microbial processes below ground. And so I like to think of carbon as the most critical currency for energy exchange in our terrestrial ecosystems. So when we really think about how energy comes to be in the soil, it's through the process primarily of photosynthesis. So plants photosynthesize, they capture CO2 from the atmosphere using energy from the sun. They build it into their own biomass, and they bring it into the terrestrial system. Microorganisms feed on that plant carbon. And so whether that be root exudates from a living plant or from plant debris, microorganisms then eventually transform that plant carbon into soil organic matter through processes of decomposition. So if we think about soil carbon cycling at a detailed mechanistic level, things get pretty overwhelming quickly. Soil carbon cycling is strongly controlled by abiotic factors, things like temperature and moisture. Microbial metabolism itself is really truly quite complex. But if we distill it down to basic principles, the amount of soil organic matter that we are storing at any one time is really just the balance of inputs minus outputs. And so if we think about how we can tweak the amount of soil organic matter storage in our landscapes, we need to either increase the amount of incoming carbon or decrease the amount of outgoing carbon. So we would want to focus then primarily on plant carbon, improving plant growth, plant biomass production and return to the soil, and decreasing outputs through microbial respiration, root respiration, or in the case of today's talk, thinking a little bit about the importance of erosion and the physical transport of soil within the landscape. So as a microbiologist, I think a lot about these things. Microbial decomposition is really a cornerstone of my research program, pardon me, thinking about how we might use management to tweak or nudge microbial processes in a way that we can store more soil organic matter. So in the last decade or so, we've had some strong shifts in our perspectives on how what the controls of soil organic matter really are. And so traditionally, how much soil organic matter was lost to the atmosphere as CO2 was thought to be strongly controlled by things like temperature and the persistence or the recalcitrance of the plant carbon sources. In terms of carbon retention in the soil, we talked a lot about humus. So the formation of stable humus providing soil carbon sequestration potential. But these shifts in our sort of fundamental thinking about how organic matter is formed have shifted toward more focus on microbial processes. So now we're really beginning to think about how microbial accessibility, so that is the ability of microorganisms to physically and biochemically access carbon in the soil, impacts how it's flowing through the terrestrial system. So rather than strong focus on soil carbon sequestration, which implies locking it away, I like to think about soil carbon cycling as a flow. So we want to continue having that carbon and that energy flow through our terrestrial systems so that the microbial processes can benefit from those energy flows, but we might nudge them such that we can actually leave more carbon behind in persistent form. So again, just setting up sort of the story about erosion that is to come, we need to stop and think about the factors that come into play in terms of how soil organic matter is actually formed and how that lends itself to lends soil organic carbon to its stability. So not all soil organic matter is created equally, and I really like this diagram by Francesca Catrufo and Jocelyn Lavallee out of the University of Colorado, where they talk about these two functionally relevant pools of particulate organic matter and mineral-associated organic matter. These pools are simple to isolate in a scientific research context, but they lend themselves really well to describing how soil organic matter is functioning and allow us to begin to predict how it might behave. So particulate organic matter you might think of as organic matter that is still somewhat recognizable as plant matter, but it is partially decomposed. And so that's depicted on the left-hand side of your screen. It lasts 1 to 50 years, so it has a relatively short turnover. Its density is lower, and so therefore it's lighter. It's more complex than the other pool that we'll talk about in a minute, and it tends to have a wider carbon-nitrogen ratio that is a slightly lower quality than mineral-associated organic matter. So this mineral-associated organic matter, or MAOM in contrast, has a slower turnover. It can last, 10 to up to 1,000 years. It's heavier, it's less complex because it's sort of either come from a simpler pool. You might think of soluble organic carbon exuded from plant roots being directly stabilized on mineral surfaces, or the other important process is microbial byproducts, or indeed their necromass, so dead microbe carcasses, if you will. And so that MAOM has a lower carbon-nitrogen ratio or a higher quality. And the things that protect these two functionally relevant pools of carbon are quite different. So thinking about how soil organic matter formation comes to be, this shift in thinking has really lent us to a filter whereby the persistence of soil organic carbon or soil organic matter is governed strongly by microbial accessibility. So what makes it accessible in contrast, then, we need to think about what are the factors that lead to soil organic matter destabilization. And so about 5 years ago, Vanessa Bailey and colleagues wrote a nice paper talking just about these destabilizing factors. And they sort of broadly categorized them into three main destabilizing factors. That is the desorption from minerals, which can be facilitated by changes in soil pH or increased soil moisture, which also in turn solubilizes additional carbon and changes the pH of the soil solution. Something that's really relevant to management is the release of carbon from soil aggregates. And so this results, of course, from tillage, physical disturbance of the soil through tillage, whereby we're breaking up the soil structure, breaking open soil aggregates, and making primarily that particulate organic matter accessible to microorganisms. So whereas previously it might have been physically protected in a soil pore where microorganisms couldn't physically get to it, if we break apart that aggregate, we're now exposing that carbon to microbial decomposition. Other naturally occurring processes like freeze-thaw and wet-dry cycles, along with bioturbation, so we might think of earthworm activity or even gopher or badger activity even, disturbing the soil also releases carbon from soil aggregates. And the final mechanism of destabilization is increased biotic metabolism. So these are things like changing the quality of the plant litter, the quality of the soil organic matter, or shifting microbial carbon use efficiency. And we'll come back to that later. But what's important about all of this background information for today's talk is that erosion, of course, is a powerful destabilizing factor. So erosion through its transport of soil across the landscape inevitably strongly disrupts the physical structure of the soil, breaking apart aggregates, exposing that particulate organic matter and even some mineral-associated organic matter to decomposition, and therefore it often leads to a loss of soil organic carbon. So we've got pictured here some wind erosion. So across the highway, there was a freshly tilled summer fallow field during a dry period a number of years ago south of Rosetown. And what we can see is that we've got significant wind erosion where we had transport across out of the field and into the road ditch. Likewise, the bottom picture shows some really strong water erosion. So if we think about then hillslope erosion, historically, we had a number of decades where annual cropping and intensive tillage really led to high susceptibility of hillslopes to water and tillage erosion. So this is a photograph of a hillslope that comes from the Soils of Saskatchewan website with soil science alumnus and PAG Mike Sulahub proudly standing at the bottom of this hill. And what we can see is a pretty strong depiction of water erosion, whereby you would have the movement of soil from the top to the bottom of the hill. Contributing also to hillslope erosion was the actual physical movement or dragging of soil by tillage equipment. So it used to be that we had less focus on contour tillage, and so that really, again, exacerbated both the physical movement during tillage operations, but also the susceptibility of water erosion on hill slopes because the water was running down the furrow rather than having to make its way energetically across the furrows as it would with contour tillage. So some work in the early 1980s when in Saskatchewan, when we were still sort of in this period of relatively high rates of summer fallow and intensive tillage by Dr. DeYoung and colleagues here in the soil science department, estimated that eroding knolls across 5 different basins were losing or had lost about 20 to 60 kilograms per meter squared. So that's equivalent to 200 to 600 tons per hectare of topsoil off of these eroding knolls. In turn, that soil goes somewhere, typically landing for the most part in the lower slope position, where they could also account for a gain of approximately 250 to 80 tons per hectare. So that tells us that not only knolls were losing topsoil, but that perhaps there was some mid-slope loss as well. And so this work was done using a tool called cesium-137 tracing. So this goes back to nuclear weapons testing, whereby the radioactive fallout from those nuclear weapons settled on the soil surface. Once that weapons testing ceased, it became a tracer for soils that had been formerly at the soil surface and were subject to erosion and transported across the landscape. So a very effective tool for tracing what where former topsoils might have been moved due to erosion events. So if we think about this process of hill slope erosion, there's sort of 3 steps that occur. First, the detachment of the soil that is being eroded, typically from the knoll. This process inherently breaks up the soil physical structure, breaks open those aggregates, and exposes some of that formerly protected soil organic carbon to decomposition by microorganisms. That detached soil is then transported downslope and eventually deposited in the lower slope positions. What's important to think about is that over time, initially upon, for example, cultivation of a native grassland with tillage and summer fallow, we would see detachment of those organic carbon-rich soils transported down the slope and deposited in the depressional position. However, if that erosion doesn't cease to occur, what eventually happens is that, of course, erosion continues, and we're now moving perhaps B and even sea horizon materials downslope and depositing them. So what we end up with is this situation of depleted knolls where we've taken the carbon-rich soil organic matter or carbon and soil organic matter rich topsoils that have developed previously under different circumstances, and we've moved them and deposited them in the depositional position. However, what we need to consider is that in those depositional positions, we had a typical soil profile to begin with, and what we've effectively done is sandwiched an inverted soil profile on top of it. This results in a band of carbon-rich buried surface horizons, both the original surface that was in the depressional position, as well as this high carbon topsoil that was originally deposited from the knoll. So it's important to recognize that this erosion process of movement of carbon-rich soils from the knoll to depositional positions is very ubiquitous in hummocky landscapes. And so my interest in this work began quite a number of years ago when I was talking to a colleague, Bert van den Bijgaard, who's an Ag Canada scientist in Ottawa. And he was approaching this problem as a researcher that works on carbon accounting for our national carbon inventory, recognizing that we currently, or at the time, we're estimating soil carbon stocks based on a homogeneous distribution of carbon across the landscape. But of course, we know that that's not the case. So Bert's work on this project looked at 6 different sites across Canada, from Alberta to Prince Edward Island. Here we've got our Saskatchewan site out at Saint Denis. And what you can see, although the print is quite small here, is that the knolls have very shallow AP or plowed horizons for the most part. And in depositional positions, we see very deep A horizons where carbon has accumulated. In those depositional positions, they did a carbon accounting, and this graph shows the topsoil stock in the white bars and the carbon stock below the present topsoil stock in the blue. The take home here is really just that this deposition of material, across the country at these 6 relatively different sites led to the significant burial of organic carbon-rich soils from upslope. So in talking to Bert, I got kind of curious about this because I thought, what is it that over the course of a number of decades leads to this retention of that carbon-rich material at depth? So why don't microorganisms simply utilize it as they would at the surface? Are there biotic or abiotic constraints perhaps? So temperature or moisture or oxygen availability? So the first thing that we did was we brought soils from those depositional positions collected at the soil surface or in the buried horizon in green and blue here. Sorry, at the soil surface in blue and in the buried horizons in green. We brought them back to the lab and we essentially removed all of those abiotic constraints by incubating them under ideal moisture and ideal temperature for about 100 days. And we looked at the amount of carbon that was respired due to microbial decomposition during that time. And so what you can see here is that the amount of soil microbial carbon respiration was much higher in the surface soils compared to those soils that had been buried for a number of decades. So this tells us that there has been some biological stabilization, or likely biological stabilization at depth, because when we simply get rid of any potential abiotic constraints of oxygen availability, temperature, and moisture, that carbon sticks around. Important to note that this carbon mineralization has been normalized to the soil organic carbon concentration. So it's really CO2 per molecule of carbon that's there. And even under those conditions, we still see far less loss in these soils at depth, indicating that it is really quite persistent carbon. So as a microbiologist, this certainly piqued my interest at the time. Hannah Conchu, who was a new master's student coming to work with me, agreed to work on looking at microbial community profiles in these buried horizons. So we honed in on the Saskatchewan site at Saint Denis, just 40 or so kilometers east of Saskatoon. And we did some microbial community profiling on the first three of these uh, profiles along the hillslope, pardon me. So this is a picture of Bert and I taking soil samples in that depositional pit. And here you can see an actual photograph of the depositional pit. What I hope you can see is that there's a very clear differentiation of some dark black carbon-rich soil about half a meter deep. So we have the original B horizon, what's very likely the original A horizon. And then, as I mentioned a few minutes ago, effectively an inverted soil profile sandwiched on top. So this part of the buried A horizon would represent what was once the original carbon-rich material on the knoll. So the first thing that we did was measure the organic soil carbon concentration depicted in green here, and also profiled the microbial community abundance indicated by the blue bars using a tool called phospholipid fatty acid profiling. And what you can see is that there was abundant microbial abundance. These are viable organisms at depth where there was lots of carbon. So this tells us that that buried carbon, although somewhat biologically persistent when we incubated it in the lab, under field conditions is supporting a viable microbial community. We then went on to profile the microbial community using DNA fingerprinting. And what we can see here is at 5 centimeter increments in that depositional position, leading from the soil surface down to 80 centimeters, we see very strong shifts in the microbial community composition. So this is telling us, okay, we've got this buried carbon-rich material. If we bring it back to the lab, it seems to be very resistant to biological breakdown, but it's still managing to support abundant microbial biomass. And that microbial community's composition has shifted in response to conditions, at 50 to 60 centimeters deep in the soil profile. So thinking back to some of the basics of how you might get soil carbon stabilization at depth, we have ruled out simple principles of abiotic constraints by bringing the soils back to the lab and demonstrating that soil carbon was relatively persistent. We did some additional experiments that I won't get into today, but we incubated those soils and added readily available nitrogen just to test, perhaps this is a nitrogen limitation. That also did not result in accelerated decomposition of the buried carbon. And Hannah did another experiment where she actually either incubated intact cores of that buried material or sieved soils just to check whether physical disturbance might accelerate the loss of that persistent carbon, and it in fact didn't have an effect. So this begins to tell us some things about how that carbon at depth might be stabilized. So when we think about decomposition, and this would represent a theoretical model of fresh carbon decomposition, we have initial fresh material that gets, on a matter of days, decomposed by microbial biomass, and the efficiency of microbial carbon use is about 50-50. So we can see, we've got this approximate 1/3 of the initial crop residue carbon. Half of that gets built into microbial biomass, and about half gets respired away as CO2. Over time, what's left in that initial crop residue becomes less degradable, and we see, we still are getting about the same proportion built into microbial biomass, but a much higher loss of CO2. And this is indicating lower carbon use efficiency. So as microorganisms decompose increasingly recalcitrant material, they do so less efficiently. Less gets built into their biomass and more gets respired away as CO2. And so over the scale of years, what we end up with, if we have a static pool of incoming carbon, is a smaller microbial biomass. We end up with persistent soil organic matter or soil organic carbon that has been physically or chemically stabilized back to those pools of particulate and mineral associated organic matter. And a huge proportion of the original crop residue carbon has actually been lost as CO2. So these buried soils with their high persistence really made us start to think about, okay, we know that we've got an active microbial community at depth, and we think that repeated turnover is leading to persistent soil organic carbon. So from basic science principles, this was really quite interesting because it sort of supports that new shifting paradigm about how we think of how soil organic matter is formed. So I developed this conceptual model, sort of hypothesizing that we might use these larger volume, high carbon concentration, high carbon persistent buried soils as a model system for understanding how we might be developing persistent soil carbon, let's say in a pore within an aggregate. On much smaller scales where measuring those processes in situ is very, very challenging due to the small scale that the processes are happening at. So hypothesizing that over time, this repeated turnover increases the amount of energy required to further break it down, the activation energy, that it could in fact deplete the energy density in what's left, that organomineral association is likely to have stronger influences in its persistence. But with taking us back to this model of particulate and mineral-associated organic matter, something that we need to stop and think about is the role of texture. So with Hannah's work, we had seen at one site that we had this very persistent carbon developed in the buried A horizon materials. But we wanted to sort of question, okay, that was a nice loamy textured soil. What happens if we have higher clay content, therefore more potential reactive mineral surfaces to stabilize this mayom? Finer textured, heavier soils tend to lead to greater aggregation. And so what that leads me to then is Amanda Mitchell's work. And so this recent work involved looking at 5 sites across Saskatchewan, starting in the southwest corner near Swift Current and extending northeast all the way up to Gronlid. And so we targeted soils with different soil textures intentionally to sort of look at how that might play into stabilization in terms of particulate and mineral-associated organic matter and microbial turnover. So we did this work in 2 different stages. We first did a field survey of soil properties and microbial communities. And then we brought those soils again back to the lab and did some further study to try to get at some mechanisms of carbon stabilization. So if we look at our 5 sites, we had sites in the brown, dark brown, black, and gray soil zones. The buried A horizon depths were approximately 40 to 60 centimeters deep, so around a half a meter. And what's important to note is the texture. So we had at one site quite high clay content, 35 and 40% respectively, and about 60% sand at the sites that were further to the northeast. I should mention that throughout the talk, the upcoming slides, I'll refer to the surface horizon as the AH, the typical AH horizon, and an AHB just denoting what was formerly an A but is now buried. They're denoted with the B. So Amanda went to these 5 sites. She took intact soil cores, she laid them out in the field and visibly identified the volume of that soil core with by color, the darkest black color, indicating where we think that buried A horizon is. So I can't recall specifically which site this was from, but you can certainly see the distinction of the original modified B horizon, the original A, the inverted soil profile that was deposited sort of over time. And what's important to think about is in these depositional positions, since that moment of original, when the soil, whether it be the original A horizon or the B horizon from the knoll was deposited, it became a surface soil. So surface soils are where we see the highest plant carbon inputs. And since that time, we've really been developing a reasonably high organic carbon soil at the surface because we know that in lower areas of the landscape, moisture is rarely limiting and we see a lot of biomass production. So looking at a cross-section of these soil cores, we can see this was the AH, the surface horizon in a depositional position, the AHB or buried AH horizon, and then the BMP. So they're or BMB, pardon me, the modified B. So quite visibly distinct. And here we really wanted to get at the question, why do high concentrations of surface-derived carbon persist at depth, despite the presence of high microbial biomass? So the next series of graphs will begin at Swift Current and over at the northeast in Gronlid, so sort of the extension of our sampling gradient from southwest to northeast. And the graphs are colored where we have the AH horizon in green, the AHB or buried horizon in blue, and then that modified B in gray. So this first figure is looking at total organic carbon concentration. And what you can see is that at most sites, we have a high concentration of organic carbon in that blue buried A horizon. Much higher at Saint Denis than Swift Current, for example. And part of the reason for this, we think, is that at some sites, Swift Current was a good example, we just had one hill slope contributing to the erosion event. But at the Saint Denis site, it was actually the convergence of 3 different eroding hill slopes. And so there was a stronger influence of erosion and deposition at that site. Looking to the bottom graph, we used, again, that phospholipid fatty acid profiling, which is an indicator of viable microbial abundance in our soils. And so we can see certainly that it was highest at the AH horizons, or in the surface, where most of the incoming plant matter is being contributed. But we also see significant microbial biomass in that buried A horizon. We looked at community fingerprinting using that same phospholipid fatty acid tool. And here we see a strong indication of shifting community structure with depth. So the AH horizons at the surface over here in green, the buried AH and the modified BM in blue and gray. And so this tells us that the microbial community structure in those buried AH horizons, despite having high carbon concentration, looked a lot more like the BM communities than they did the surface communities. So similar to Hannah's DNA profiling work, we see a significant shift in community structure in those buried horizons. We looked at here the mineral associated and particulate organic matter pools. So what this is telling us is the proportion of total carbon stored as mineral associated organic matter or particulate organic matter. And what we can see is that in both the AH in green and buried AH in blue, we had higher, or about 75% of the organic carbon being stored as mineral associated, and about 25% being stored as particulate organic matter. We also see that there was significantly higher mineral-associated organic matter, a significantly higher proportion of carbon stored as mineral-associated organic matter in those buried A horizons, sort of along confirming our hypothesis that mineral association as a protection mechanism would be more important under those prolonged burial conditions. We also looked at the proportion of soil organic carbon in the AH horizon and buried AH horizon that's stored as microbial necromass. So this is thought to be a really important pool for persistent carbon. Microorganisms like to live on surfaces, so you can imagine they might really enjoy their existence living on a reactive mineral surface. And it turns out that at their eventual demise, their necromass or their corpse is still attached to that mineral surface. And so it makes a lot of sense that that microbial necromass could perhaps be preferentially protected in the soil. And so we can see that there weren't huge differences, about 10 to 15% of total soil organic carbon stored in this microbial necromass. But in our sandy buried soils, there was actually less microbial necromass. And this is because, of course, sand particles are not reactive. By being sandier, that means they had less clay and so less capacity to protect that organic matter. So that leads us to, we've sort of characterized Amanda's 5 sites. Soil texture seems to be having some influence on how carbon is being stored in surface and buried horizons. We wanted to just confirm that at these new sites, we would see that same biological persistence or biological stabilization if we brought them back to the lab and we incubated them there. So this data is a 100-day incubation of the surface horizons compared to those buried AH. And what we see again is that the potential for loss through decomposition, measured as CO2, is greatly reduced in those buried AH horizons. And so this represents, how many CO2 molecules are lost or respired away per every carbon molecule present. So normalized again to the carbon concentration and much, much more CO2 susceptibility to decomposition in the surface soil. So we do again see this persistence and stabilization in those buried AH horizons across all 5 sites. The last series of measurements that we made is using a technique called isothermal calorimetry. And so this is a really interesting way to sort of look at the efficiency of microbial decomposition. So this is a picture of the microbial calorimeter. What it does is measure very, very small heat fluxes over time. It was, just to sort of contextualize, originally developed by TA Instruments in conjunction with a chemical company to understand or to be able to track the declining metabolism of a single mosquito in a vial dosed with pesticide. So that's how sensitive it is. You have a mosquito living in this vial, you dose it with pesticide, and its declining metabolism can be tracked through a decrease in heat production. So a very, very sensitive way to measure the amount of heat flow in a vial. So what we're really doing here is putting a small amount, 5 grams of soil, sort of packed to a very specific bulk density that represents what it was in the field, into these 20 ml ampules. We pre-incubate them to make sure that we're putting them in there at a steady state. And we then measured the heat flux during either decomposition of the indigenous soil organic matter or heat flux when we added glucose, which is a very readily available sugar source for microorganisms. So just sort of very readily available carbon. It's a short-term assay. It lasts only about 48 hours because the assumption is that we need to utilize all the glucose that we've added. But the principle is that the more heat released during metabolism, the less efficient that metabolism is. So if there is an energy content of food, we want more of that energy to be retained in the microbial biomass than to be wasted as heat. So thinking about the basal or background soil organic carbon thermodynamics, this chart, again, we've got the AH horizons in green and the buried AH in blue. And here we've got the heat flux per CO2 respired. So it tells us essentially how much heat is produced per microbial respiration. And we can see that for the most part, it was higher in those buried AH horizons. And this makes sense because we would expect that persistent carbon takes more work to metabolize for the microbial community. And so in taking more work, they're less efficient at it. They need to produce more complex enzymes to break it down. And so it's quite sensible that we would see this increased heat per CO2 in that persistent carbon. However, we saw a pretty different pattern when we added readily available glucose, in that the buried AH horizon soils seemed equally able to use that carbon efficiently. And so while there were some differences in our sandy soils where there's not a lot of protective mechanisms for the indigenous soil carbon, generally we saw that there was equal efficiency of these buried AH and surface horizons in terms of using readily fresh incoming carbon. The very last thing that we measured was thermodynamic efficiency. So you can sort of think about this as the heat lost from glucose metabolism relative to the total energy that was contained in that glucose. And the take-home message here is that really thermodynamically, those buried AH horizons were actually more efficient at squeezing out the energy contained in that glucose than the AH horizons were. So thinking back to this idea that we now have perhaps a more thermodynamically and shifted community structure in the buried AH horizons that have a lot of indigenous carbon, we can begin to sort of try to relate this energy use efficiency to soil properties and microbial communities. So we did regression analyses that just sort of allowed us to relate some of those thermodynamic properties. So straight up basal respiration, basal heat production, that heat per CO2, and finally the calorimetric efficiency of glucose use to different soil properties. So we've got texture, total organic carbon, particulate, and mineral associated organic matter, as well as necromass. And the take-home message of this particular slide, we won't get into the details too much, is that we were somewhat surprised to see that there was very little relationship between the thermodynamic efficiency and organic carbon, soil organic matter properties, like the proportion as necromass or the proportion as POM or MAOM. However, when we looked at the relationship between microbial community properties now, so these are the NDMS axes. So I'm just going to switch back a couple of slides. So this is essentially the score on this axis with depth and the score on this axis with depth. So an index of community composition. What we see is very different relationships in the AH horizon of thermodynamic properties with the microbial community compared to the buried HB. Likewise, we saw different patterns of the relationship of energy use efficiency with the microbial community structure in the AH and AHB horizons. So that got pretty deep into mechanisms of soil organic carbon concentration, but to think about some sort of take-home solution or conclusions of the basic and applied science of this work. So what we discovered here was that soil organic matter turnover, or the persistence of soil organic matter, was linked to different soil and microbial characteristics in the buried horizons compared to surface horizons that may have had very nearly equal carbon concentrations, but different soil organic matter composition. We confirmed that those buried carbon-rich soils had an active microbial community despite, being buried at a half a meter deep. And what we saw was that repeated turnover in those buried AH horizons seems to lead to persistent soil organic carbon. So this supports sort of shifted thinking in how we, in the theoretical models of how persistent soil organic matter is formed, that it's less about the recalcitrance of the original material and more about repeated turnover, whereby at each turnover event, the microbial community increasingly squeezes the energy and the easily available carbon out of that material, leaving behind something that is more persistent and perhaps shuffling it into pools that are also more stabilized, like mineral associated carbon, things like microbial necromass. So coming back to this conceptual model of using these buried carbon soils as a hypothetical model for studying, or as a theoretical model for studying things that might be happening at a scale that are very difficult to measure, like in a pore of an aggregate or on the surface of a bit of particulate organic matter locked away in an aggregate, it prompts us to think about, we added glucose to our soils and saw higher efficiency of its use than we did we saw higher efficiency of fresh carbon use in those buried soils than we did in the surface soil. So what does that mean for new incoming carbon to these buried AH profiles at depth? For example, leached soluble carbon or root deposited carbon. And so this allows us to really begin to think about, okay, if we want to nudge our inputs minus outputs to have more soil organic carbon storage, can we query these places, these special places in the environment where microbial carbon use efficiency is actually higher, to understand processes going on at the very small scale in typical surface soils through a larger soil volume in these buried horizons. So scaling out to sort of more applied applied relevance of this work. We know that in Saskatchewan, widespread adoption of conservation tillage and the essential elimination of summer fallow has drastically reduced hillslope erosion. However, we're left with this legacy of eroded knolls. And so I think Dr. Jeff Shano was talking to you yesterday about some work that he's done with amendments on knolls. And he mentioned Dr. David Lobb at the University of Manitoba, who does quite a lot of work in this space. So I would just point you to a really nice paper that was recently published by that group. And so they sort of assessed the proportion of Saskatchewan cropland, or actually national cropland, at high erosion risk between 1971 and 2015. And so it's really encouraging to see that in 1971, 36% of our cropland was at high erosion risk, and that number has been essentially eliminated through these conservation practices. However, very impactful is that their estimation of lost revenue through reduced crop production was approximately $7.5 billion over that time. And so this comes back to that legacy of low productivity on highly eroded knolls. We're still seeing the impact of this carbon loss and deposition. So there's been some work to think about, can we take carbon-rich soils from the depositional position and actually transport them back up to the knoll as a way to accelerate productivity in those places. So again, collaborative work by Dr. Dave Lobb and Curtis Cavers in Manitoba did some work in Minnesota, and they showed that 15 centimeters of topsoil replacement on eroded knolls increased soybean yield by anywhere between, or soybean and corn yield anywhere between 12 and 59%. What's important to think about is it also increased biomass production, even on moderately eroded land. This is important because if we go back to that how to build soil organic matter and how to restore fertility, we really need to focus on increasing plant carbon inputs. So anything that can allow us to jumpstart productivity on those knolls is going to increase that incoming carbon. It therefore provides energy, fuel for the microbial community to begin building and accelerating soil organic matter accumulation, which of course we know is the cornerstone of soil health. So Dr. Henry Jansen wrote a really nice paper a number of years ago where essentially he talked about this idea that soils have memory. So if we look at this picture, this was just a field on Highway 14. I took this photo about 5 years ago on one day in June. And I hope what you can see with the contrast on your screen is that we can see a lighter color in the knolls. That might be partially contributed by drier conditions compared to the lower places in the landscape. But this is something that we don't often see. We don't often see summer fallow in Saskatchewan anymore. And so it's really very apparent that even in this pretty gently undulating landscape, there is this legacy of past tillage and water erosion where we've got depleted fertility on the knolls. And if we focus on sort of thinking about how we might use principles of soil organic matter formation and microbial activity to think about restoring them. Certainly, transporting topsoils to the knoll is one option, but we need to think about what we might be exposing in those depositional positions where we've removed that surface layer. And so those are some of sort of the next questions that we're thinking about with this work is what happens, on the landscape scale if you expose those buried AHB horizons. Do they behave and support high fertility and crop production, or does that increased persistence of carbon lead to changes in function with respect to cropping objectives? So with that, I'll just wrap up. I want to sincerely give so much credit to the very talented Amanda Mitchell, who recently finished her master's here at the University of Saskatchewan and is starting a PhD at the University of Guelph, to my research team who contributed in a myriad of ways to this work, and to past Ag Canada and USAS collaborators. And thank you very much for the opportunity to come and talk to you about this work today.
Thank you so much on this. Some of the questions had been answered in the presentation. So one of them had been about moving what happens to buried soils microbially and chemically when moved again to the hilltops. Now my question, kind of elaborating on that, is how are we doing that? Are we doing a deep tillage and trying to move the soil physically? Are we looking at just again, the next question has a microbial profiling. So are we taking some of those microbial profiles and then inputting those onto the hilltops, or how are we going to change some of that dynamic?
Yeah, and so I think, as I mentioned, Dr. Shana was talking about this a little bit yesterday, slightly different context where he was using replaced topsoil as a control to compare for other amendments. Sorry, I've got your screen over there, so I'll try to look here. And Dr. David Lobb has really been doing a lot of work in this space. So typically what we think of as an actual excavation of soil from the depositional position and physically moving it to the knolls. Getting it to stay there comes with a myriad of challenges, but certainly, I mean, it's not only organic carbon that imparts that fertility and that productivity. And so by moving those depositional soils, you're moving sort of all of that inherent goodness in those highly fertile soils from the lower landscape position back up to the knoll. And so I think long-term implications for fertility and productivity at both of those positions are an ongoing research question.
Are you guys looking at some profiling of the microbial biome and using that as some soil health interventions?
So we have not been thinking about, for example, taking a thin layer of those depositional soils and using them as an inoculant. I guess my perspective is if, in terms of microbial community health, if you build it, they will come. They ultimately feed on plant carbon. So if you can get plant establishment on those knolls and return a lot of carbon, that can be a big challenge because soil structure can be poor, root infiltration can be an issue. We know they're always drier than the rest of the landscape because they're water shedding. But if you can build a carbon food source there, it's unlikely that you would need to re-inoculate necessarily. I think that those downslope soils, you would see a restoration of a healthy microbial community if you bring the food. Balanced fertility through input use is really important, of course, as well.
We talked a lot about growing forages for salinity and also in our poorer producing areas. Would growing forages on eroded knolls help increase carbon and organic matter faster than manure or other soil amendments like replacing eroded soils?
Yeah, I think if you can get certainly perennial crop established, I mean, there's plenty of evidence that root carbon sort of feeds that system more efficiently than above ground carbon and might even perhaps get shuffled and nudged into those more persistent pools. So I think if you've got a particularly leguminous forage that can bring nitrogen into the system as well, you'll have some good capacity to jumpstart productivity and microbial carbon processing and soil organic matter building in those knoll positions for sure.
And when we're talking microbial profiling, can you roughly estimated beneficial versus pathogenic microorganisms in the soil? And how accurate are the databases?
That's a good question. So I think, I mean, there are microorganisms are kind of wild, right? Because you can have an organism that is beneficial in one context, and if you change, even just in time, the situation, it can become a pathogen. And so there's plenty of examples of that in like common microorganisms that commonly occur in our healthy soils. So usually pathogen load is low unless you create a circumstance where it's got an ecological advantage. So continuously using the same crop is one way that can happen, of course, right? If you continually provide a host for a pathogen, you can build up that pathogen population and end up with a disease problem. So again, sort of speaking to the importance of using crop rotation as a disease break, but I also like to think of it as using it as a diversified diet for the microbes. So just like us, microbes like a diverse diet, a healthy, balanced diet. And so one really terrific way to suppress pathogens or to prevent them from sort of getting an edge in your soil is to just mix up and have diverse rotations.
Okay. How might climate change affect the stability and turnover rates of soil organic matter?
Yeah, so this is something that's under some pretty intense study. I mean, we can think about climate change as having an increased temperature during the non-frozen season. And so that certainly does have an impact on the rate of decomposition of crop residues. We've got a really beautiful, cross Canada study showing that we can do a very good job of actually predicting how much crop residue might be retained by simply looking at the cumulative days above zero, if you will, or cumulative thermal time, we called it. You could think about climate change in terms of the increasing frequency of intense weather events. So that might increase the susceptibility or the likelihood of big erosion events. And I think Dr. Bulch talked about that earlier today. You might also think about climate change in terms of increasing CO2 in the atmosphere, perhaps increasing photosynthetic rates and generating more net primary productivity. So I think I always think about it in terms of inputs and outputs. And so what we can do with management, we ought to do because climate change is something that needs to be mitigated at a different, well, certainly on farm, we can make our contributions, but in terms of reactivity to changing temperature, for example, we're going to be looking at climate change mitigation or adaptation, pardon me.
When you're looking at some of these, have you looked at the economics of doing your organic matter additions, such as manure on the knolls, instead of digging out buried topsoil and putting it back?
I haven't. So I'm as a, to stay in my lane, I'm a microbiologist, and so I tend to think about things at the process level. But I do think looking to Dr. David Lobb's work at the University of Manitoba might be a good place to find some of that information. The paper that I talked about at the end of the presentation today didn't look at alternatives. And I don't think Dr. Shano is looking at that. I know that manure was one of the amendments that they used on the knoll, but I don't think that he's looking at the economics of that as well, or either, I should say. But there may in fact be work out there. I'm just not aware of it.
And then the last one here, these might be fighting words. Like our previous speaker discussing farming around a pothole for biodiversity reasons, not farming the knoll tops would save money instead of productivity losses with erosion, and now the cost of moving soils back with oxidation risks of the moved soils. Do you have any comments on that?
Um, Again, I mean, it's out of my wheelhouse. I think if you took knolls out of production but had them vegetated, you might find that you could provide other ecosystem services. So if you had a diverse plant cover, for example, you might see them providing, those additional ecosystem services like being biodiversity hotspots, for example. Anytime you've got photosynthesis, you will be supporting a below ground microbial community, and that in turn has a regulatory function with respect to nutrient turnover and cycling. I mean, I think that's an individual producer decision. Can you do it? Is it feasible logistically? Is it feasible economically? You're taking land out of production. Can you even just move around it, right? Like as our implements grow in size, those kinds of decisions are becoming increasingly complex. So.
Thank you so much for being with us here today and giving us lots to think about on carbon sequestration and organisms in the soil. It's nice to think of things deep down, not just right on the soil surface. So thank you so much for being with us today.